We think that lipoxins have have effects on multiple different cell types that are involved in atherosclerosis and indeed in diabetes-associated atherosclerosis. So we know overall that they can cause a reduction in plaque burden, and we know that that's as atherosclerosis develops, and we also know that when we add them to establish plaque models or to establish atherosclerosis and diabetes, that they're able to regress the disease. And this is work that we've carried out in collaboration with Professor Mark Cooper and Phil Cantheridis in Melbourne. So we know then from parsing the different cell types involved that lipoxins have distinct effects on macrophages, on endothelial cells, on vascular smooth muscle cells. So it's sort of a combined both an anti-inflammatory and a pro-resolving activity. Well, firstly, I think we've made colossal leaps in the last 2 decades. It's been slow, but we're gathering momentum, and there's absolutely greater acceptance of the idea that resolution is an active and a physiological process. I think as we understand more of the molecular drivers of these processes, we're able to think of ways of devising mimetics, so compounds that can mimic the endogenous agonists of resolution. And so in the case of the work that we do in University College Dublin, in my lab and in collaboration with a chemist, we've been able to make synthetic mimetics of lipoxins, and we have shown that these have efficacy in numerous preclinical models. And so I think we need to understand their pharmacokinetics and their pharmacodynamics a little better, but certainly their pharmacokinetics. And I see no reason why we shouldn't be able to make significant progress in the next 5 to 10 years, to be honest. I think it's time to really, address the issue of resolution as opposed to blocking inflammation, because we know that there are numerous negative effects of anti-inflammatory compounds, but our pro-resolution compounds are very specific, and probably most importantly, they're very potent. And by virtue of their potency, one has to presume that pharmacologically they will have less off-target effects, or at least that would be the conventional wisdom. So I would say we're pretty optimistic about developing these molecules. We have numerous data from preclinical models of acute inflammation, but probably even more impressive are the data with the people we work with in Melbourne with Mark Cooper and Phil Cantheridis in the vascular complications of diabetes, which are really difficult. They're difficult diseases to model because they're chronic complications, and they're also very difficult to treat. So we have some really nice data which we've developed with Mark and Phil in Melbourne that I think are going to be very instructive, both in terms of the molecular mechanisms and in terms of potential protection. Yeah, there really aren't, certainly for diabetic kidney disease, which we have to accept is actually one of the, a major complication of diabetes. We're all more than aware that diabetes is a global problem, but we never really think that actually it's not diabetes per se, it's actually the complications of diabetes that are such a big challenge, and particularly diabetic kidney disease. So this affects probably 30 to 40% of people. It's a chronic complication. And actually, it's the one thing that's most associated with premature mortality. So it's a really big problem. Over the last couple of years, with the increased use of different therapies, they don't actively target diabetic kidney disease per se, but they are beneficial. But we think there's absolutely room for something else in the field, like on top of standard of care therapies, something that would promote resolution. And whereas diabetic kidney disease would not have been considered a conventional inflammatory condition. By conventional considerations, we now know that both from genome-wide association studies, from gene expression studies, from biomarker studies, we know that inflammation is a key driver of diabetic kidney disease. So we think that this provides a window of opportunity to promote resolution in that context that hasn't been approached to date. I guess what excites me most is that, it's a slow process, and we've built absolutely incrementally. We started off with a curiosity about the biology of lipoxins and about how they signal and the cell types that they work in. And, this has gone on over a couple of decades, in fact. So it's been a very slow process. So our understanding of lipoxin biology from my lab in Dublin has increased a lot over the years. And then we try to apply that knowledge to models of disease, building up from cell models into preclinical animal models. And now what we've been working on most recently with our collaborators in Australia that I mentioned, and also with Professor Pat Gyrey at University College Dublin, has been the idea of synthetic lipoxin mimetics. So we now have a good idea of what lipoxins do endogenously, and now we're able to mimic them chemically. So that's the most exciting thing. And, each time we sort of nudge forward, we seem to make, little advances that are really positive and gratifying. And, we're finding out more science, more knowledge, more fundamental truths that we can apply in a context of a therapeutic context, we hope, eventually. Well, interestingly, we know from the work of many people that lipoxins and other pro-resolving mediators are really effective in regulating the acute inflammatory response. So, for example, regulating neutrophil trafficking, endothelial cell activation. But the work that actually we have been particularly driven by in my lab has been actually more of the chronic complications of disease and chronic complications of inflammation, which essentially inflammation and its resolution are really physiological responses. And if the amplitude or the duration of inflammation persists, what happens is we get, dysfunction leading eventually to fibrosis. What we've been able to see has been that lipoxins actually are antifibrotic, and that's been quite an advance. And through work that myself and my colleague Owen Brennan have done, we've been able to see that some of the mechanisms that underpin this antifibrotic or fibrosuppressive response to lipoxins. So we know that they absolutely work in acute inflammatory scenarios, and that's well established in multiple labs. But this idea that they're also fibrosuppressant is relatively new, and we know that this is driven through specific classes of microRNA and through epigenetic regulation. So that's quite an exciting field.